we want to today, we're going to take a little bit of a different focus. As you can see, we are very interested in the issue of nutrition and reproductive success and how it's impacting caribou. And much of this work was kind of began out of a paper that we just published in the December issue of Frontiers in Ecology. You can see here we fortunately got the cover photo of Frontiers, which we were very excited about. And for those of you interested, there's also a podcast on the Ecological Society of America's webpage that you can hear the work. One of the important things of this study was that we used detection dogs to enable us to really comprehensively monitor the ecosystem, taking advantage of the ability to get very fine-grained measures about population dynamics of the animals. We did a very extensive mark recapture analysis to allow us to accurately determine population size of the caribou in our study area in the oil sands. We were also able to, with DNA, to get species, sex of all individuals we sampled, and individual identity. We got a wide variety of hormones from the samples. We got stress hormones, reproductive hormones, nutrition hormones. We were also able to look at the diet in these samples and to use the sample locations to get an idea of resource selection. So we were able to, from this, get a kind of a consilience of evidence of what is impacting the caribou in this ecosystem. And this kind of set the key findings of this work were, first of all, that we found from our mark recapture analysis that population size of the caribou was actually more than double of what the current perception was for this population. And we also found that over the 4-year study, the abundance of caribou, moose, and wolf, all of which we did mark recapture analysis, did not change over that 4-year period. We found that wolves were primarily targeting deer in our study, as has been shown here and elsewhere. But what was really interesting about this was we looked at the variables that predicted resource selection of deer, and the wolves were actually also targeting those same features that the deer were targeting, and those were they were negatively correlated with the same features that caribou would prefer, which kind of implied that by going after a deer, it's actually taking the wolves out of the caribou habitat. But the most important finding, we believe, is that caribou resource selection and physiological health were very much negatively related to human activity on the landscape here. This is based on data from 2006 and 2007. And really the driver in this, we seem to think, was nutrition. So that leads me to the current study that we did, where what we wanted to do was to acquire much more detailed information about caribou winter diet and its influence on resource selection, nutrition of the animals, as well as female reproductive success. And to do that, we used 502 caribou samples collected during 2009. Now, since we're looking at reproductive success, it's very important to have a good pregnancy diagnosis, and you can see that here. This shows you progesterone levels for female caribou and for male caribou, and you can see that the females are markedly higher. These are a seasonal reproducing animal where on an annual basis, and in those kinds of ungulate populations, most animals conceive, and you can see a very distinct difference in pregnancy levels of the female versus males. Also, one of the things that happens is as gestation progresses, placenta is getting bigger, the placenta is making progesterone, so a healthy pregnancy should show increased levels of progesterone over time in females, which you see here, increasing over time. But when you look at the same relationship for males, down here, this line is shifted here to show us males, you can see no relationship. So we've got a very, very reliable measure of pregnancy in these animals. All of these animals were genotyped multiple times to be 100% certain on the gender of the animals, and another study in Scandinavia using our same antibody as this study showed 100% accuracy in predicting pregnancy. And the higher the progesterone levels, the higher is pregnancy health. And this has been shown in many, many different species. In humans, low progesterone levels during pregnancy explains 25% of habitual abortion, for example. Okay, so now that we had a good pregnancy measure, we took our 502 samples, we had scat locations, we had hormone levels on cortisol, thyroid hormone, progesterone, and I'll talk about what each of those indicate in a minute. We had DNA to get the sex and individual identity, and we took those samples and we sent them to Washington State University to the Wildlife Habitat Nutritional Laboratory. And what they did was they do a microscopic analysis of every single scat sample, and they do 25 separate snapshots of each sample to get an idea of the proportion of all the diet components in the caribou samples. And what we know from work to date, not by many different people, is that woodland caribou are foraging specialists in the winter, primarily consuming lichens, and woodland caribou have very high fidelity to caribou ranges, even when lichen availability is reduced in cases of such as fire. The first thing we wanted to do was to really make a good spatial lichen index, and to do that we essentially related caribou pellet composition in those 502 samples to a variety of spatial variables that reflected lichen habitat, as well as a fire year, because fire is very inhibitory to lichen growth, and latitude and longitude. And then what we did was we compared that to 200 independently sampled plots across our study area here. And what we have ultimately is a very broad scale index that is highly correlated with field plots and diets in the caribou. Now, what this shows you is that lichen index that we developed, and this is for various plant type parts in the diet, for lichens, Petrum and vaccinium, equisetum, and conifer. And one of the things that you see here is that as lichens are, the habitat is favoring more lichens, you're eating a very, very high percentage of lichen. But in those same habitat places, they're eating very little of the other kinds of food types. So that's very important, because what it's showing is that this lichen model is nicely predicting where we're getting lichen in the diet. And in fact, lichen was in 60% of the samples had a high percentage of lichens, which means these guys are being driven by lichen in this model. And when the spatial index suggests that lichen is low, we see other products become important, although not nearly as important as lichens, such as the Petrum vaccinium and the equisetum and conifer. Now, when we looked at resource selection probability functions of these models, we did it in two different places in our site, in the egg pony area, and then we compared it to the Waiu area. Most of our site was in the egg pony, where there was a high amount of lichen present in that area, but in the Waiu area, there was the House River fire in 2002, which really wiped out a lot of the lichen in the area. And one of the things you can see here with the resource selection is that they are very much, caribou are very much, and this is female caribou, are very much selecting areas where the lichen are most abundant. And in the area where we do not find a high density of lichen, you actually get the opposite pattern. And similarly, when we looked at the other very important variable in our model, which was the density of human use, mainly by primary and secondary roads, again, in this important egg pony area, you find that caribou prefer areas farther away from roads. The further away from you are from the roads, the more these female caribou want to be there. And in the area, the Waiu area, where the lichen was less abundant, it's much equivocal and a little more complicated to interpret. So what are the implications of these changes in diet and resource selection is really what we wanted to get at. And to do that, we use endocrine analyses that our lab pioneered in the mid-'80s and have been applied to a wide variety of different species. The first hormone that we use is thyroid hormone, or T3, is the most active thyroid metabolite of thyroid hormone. And what's important about thyroid hormone is that it responds to nutrition by lowering metabolism. When you are starving, you lower thyroid hormone to lower your metabolism, which makes you much more efficient at conserving the food that you have available. It's a very long-term strategy for controlling diet. You lower your metabolism so you use your food at a lower rate. That's, by the way, why diets don't work, is because you start to starve yourself and you gain a lot more for that one potato chip than you would otherwise. Now, the second measure that we use to look at nutrition is, and it's important to note that high is good. High thyroid hormone is a good thing. The second hormone we used is glucocorticoids. Cortisol is a common glucocorticoid that many of you have heard about. This increases in response to both psychological and nutritional stress. But glucocorticoids work by mobilizing glucose. They give you high energy immediately when you need it. It's a very short-term response to nutritional demands, because what it's doing is it's saying, I need energy now, and it absolutely mobilizes glucose at a very, very high rate. So high cortisol is bad. So it's just the opposite of thyroid hormone. And then, of course, the third measure you've already learned about, progesterone, which is a good index of the occurrence of pregnancy and its quality, including whether or not you're getting an abortion or a bad outcome. So, of course, high is good. Do I have time to take a sip of water? Thank you. So, when we correlate these hormones with the various food products that they're eating, for example, with thyroid hormone, where high is good, we can see that for Empetrum and Vaccinium, thyroid hormone nicely increases with the amount in diet, and similarly with conifer and also with lichen in the diet. Now, one of the things to know about lichen is that lichen is extremely high in polysaccharides. It's got tons of sugar in it. It's just that it's very difficult for most species to access. And so cortisol is a very important one for lichen as well. And here you see cortisol, which, remember, increases with psychological or nutritional stress. And you can see, whereas thyroid, by the way, doesn't change with nutritional stress. I'm sorry for not mentioning that earlier. And what you see here, where high is bad, is that the more lichen in the diet, the lower is cortisol. A very, very tight relationship. And we can also see this for equisetum in the model, and also, though, for distance from roads. The farther you are from roads, the lower is your cortisol, which is also consistent with our paper last month in Frontiers in Ecology. And when we link that to the thyroid hormone models to our spatial lichen index, the various hormone levels to our spatial lichen index, we see that, again, for thyroid hormone, the relationship to the lichen index is a little bit less clear. Now, partly because lichens the thyroid hormone seems to be responding more to these things that they're eating when lichen is not that available in part. And these are also foods that tend to be higher in fats and proteins, but it also shows a slight increase with the amount of lichen. But the critical thing, and that's high is good, the critical thing, though, is the glucocorticoids. So if you look at glucocorticoids with the lichen index, you see that when when you're in an area where there are low lichens, you have extremely high glucocorticoids, and as the amount of lichens go down, when you are in an area with low lichen abundance, the stress hormones are very high. As the percentage of lichens in the habitat increase, cortisol goes down, which is a good thing because high is bad. And we see, with respect to progesterone, the pregnancy marker, showing us reproductive growth, that the more lichen in the area, the higher becomes progesterone. So there is a very strong demographic signal to these kinds of events that we're starting to see. And this graph here shows you kind of how that works from a physiological standpoint. It's showing you progesterone concentrations of female caribou as a function of glucocorticoids on this axis, and then as a function of increasing thyroid hormone. And so what you see, remembering that high is bad for glucocorticoids, the higher is the glucocorticoids, the lower becomes progesterone, and you're getting to these higher stress areas where we are at spontaneous abortion levels absolutely in the caribou. And we are also at levels that tend to create a very bad pregnancy outcome, so that the offspring is likely to die afterwards. Whereas thyroid hormone, where high is good, is showing you the opposite fact. As thyroid increases, progesterone goes up. So this is a very strong nutritional signal. A nutritional signal is good nutrition, high thyroid, low cortisol. Bad nutrition, low thyroid, high cortisol, and we're seeing exactly that effect of nutrition on this important reproductive marker in the caribou. So to conclude, we tend to find in our studies that factors that influence caribou population growth. Sorry, slow down for a second here. Factors that influence caribou population growth other than wolves, actually, we believe, have been underappreciated in this system. And we've been spending a lot of time to try to bring this point forward. And obviously, it's a different perspective than is what has been portrayed here. But we're not saying it's unimportant. We're just saying there are other factors here that we really need to consider. And it's particularly important given the consequences of wolf removal to the whole ecosystem. The other thing is that lichens are very important in the system. They comprise a large part of the caribou diet. They are, the caribou are highly selective to eat the lichen during the winter time, and this selection impacts their nutrition, and it has a significant reproductive consequences. So it is a fine-grained measure of impacts on population growth in the system. So what to do? Lichens are rare in our study. It's about 5% of landscape cover. And one of the things, though, is that given how important it is, if we want to start talking about critical habitat for caribou, we got to start talking about lichen. And these lichen-rich areas indeed provide critical habitat for the caribou, and its presence enhances their reproductive success. So we think that management should conserve these lichen-rich forage habitats. It's a very important element of this, and we think that we should minimize overlap between human use and these lichen-rich habitats. It so happens that a lot of the lichen is most prevalent in these areas where you have the highest use on our landscape. And I have this bad word up there, but I didn't say adaptive management, I said adaptive monitoring. Because one of the things that you hopefully can see from this work and the previous work that we published is that we are getting a consilience of evidence going on here showing consistently the same effect, and they're fine-grained measures that allow us to look at physiological health, reproductive health, and population change on a short-term basis. So if we are going to implement these strategies, we need to do this fine-tuned monitoring year by year as it's going on to see, are we doing this right? If we aren't, we think that these are sensitive enough measures to detect it, and we can make an on-course correction. And that's what we mean by adaptive management here. And if we are going to try to reclaim habitat, we should really consider the reestablishment of lichen. And with that, I close. I want to thank Statoil Hydro for supporting this work.